for these traits and the markers linked to these QTLs, we can directly go for marker-assisted selection of a perfect recombinant in a large population. So to know the mapping, to know the QTLs, we need to go for QTL mapping. In conventional biparental linkage mapping, we need to develop different mapping populations, which may include F2 population, F2 red, F3, backcross populations, RILs, NILs, and many others. This generation of this mapping population takes a very long time. And after generating mapping population, we need to go for phenotyping, genotyping, then linkage mapping, and then we identify the QTL. After doing all this, the resolution of the QTL identify will be very poor. Why this QTL resolution is poor? It is because number of recombination events that have happened during generation of these mapping populations is very low. That's why the resolution is poor. How can we improve this resolution of the QTL? We need to use the mapping populations which have undergone several rounds of recombinations and meiotic events. So alternatively, if we can use a collection of germplasm or collection of land traces where large number of historical recombination events or mutational events have occurred, we can improve the resolution, and we can improve the resolution of QTL up to 1 centimorgan, which is far better when compared to RILs or double haploid populations. So what is the approach where we can use this kind of population? The approach is association mapping or GWAS. To know more about the GWAS, I welcome you all to my first doctoral seminar on genome-wide association studies, fast-forward approach for gene mapping. In my seminar, I'll be talking about association mapping, linkage mapping, and versus association mapping, different types of association mapping, linkage disequilibrium, steps in GWAS, case studies, limitations, and future prospects. First, let's see what is association mapping. Association mapping is linkage disequilibrium based mapping approach to detect marker trait associations. So here, the marker, we will detect a association between marker and trait, which is based on linkage disequilibrium. We'll be talking about this LD in further slides. In case of association mapping, we need not to generate a mapping population. We can directly collect the accessions or the germplasm lines, and we can directly use them in mapping. So we save a lot of time here, which is taken in conventional biparental mapping. It uses evolutionary recombinations and mutations. And because of a high number of recombinations, we are going to have a high resolution of the QTLs. Then it can be used in annual, perennial, and vegetatively propagated crops. And germplasm and breeding lines can be used for mapping. And one more advantage, which is very important, is we can tap multiple alleles. In case of conventional biparental mapping, we are using only 2 parents, and that's why maximum number of alleles that we are going to study for a trait will be 2. But in this case, we are using a large collection or large number of accessions. That's why we have a diverse panel, and we will have multiple alleles for a particular trait. Let's see what is the basic difference between conventional linkage mapping and association mapping. So in linkage mapping, we use structured population. Structured population means the 2 parents what we are using. In those 2 parents, maximum alleles will be similar, and in the mapping population, those will be fixed. That's why we call it a structured population. Whereas in association mapping, we will be using a diverse panel of germplasm. So here it will be unstructured population. Because of low number of recombination, it will have low resolution. Here it will have high resolution. Here we can use only few, specifically 2 alleles can be studied. And in case of association mapping, we can study many alleles. In case of marker density, here we use moderate marker density. Here we need high marker density. In case of linkage mapping, we can use it in annual and biennial species. But we cannot imagine developing a mapping population like F2, F2, F3. In case of perennial species, it is very difficult, or I will say unimaginable. But in case of association mapping, we can simply collect those accessions, and we can go for the association studies. This takes a very long time, whereas association mapping takes very less time. What are the different types of association mapping? There are two types. First is candidate gene-based association study. Another is GWAS. So, in what is candidate gene-based association study? If we have a preliminary study or a rough idea about a location of QTL or genes, say suppose QTLs are located in this chromosome of the genome. So we can target only that region for association study using a diverse germplasm panel. So that will be called as candidate. We will be targeting only the hypothetically derived or QTLs which are derived from preliminary studies. But in case of GWAS, we do not need any preliminary study. Markers distributed across the genome, over the genome, are employed to evaluate all the genes simultaneously. And we can use this for complex trait. We can reveal the entire pathway of a particular trait. It is more comprehensive approach and no preliminary studies are required. So I said that association mapping is based on linkage disequilibrium. So let's see what is linkage disequilibrium. So if we have, let's consider two locus. At first locus, there are two alleles A and G. Another locus, there are two alleles C and T. So genotypes, so in any population, if all the alleles from one locus are free to segregate with any of the allele from another locus, then that population will be called as at linkage equilibrium. In first case, we have how many numbers of genotype we have with AC? There are two with AC, two individuals with AT, two with GC, and two with GT. So genotypic frequency of all the genotypes is same. So all the alleles from this locus have equal opportunity, or they are free to move or segregate with all the alleles from another locus. So this population is said to be at linkage equilibrium. But if we see the another case here, 4 individuals are carrying AC and 4 individuals are carrying GT, but individuals carrying AT and GC are not available. So here allele A is anyhow not allowed to segregate with T, and G is not allowed to segregate with C. That's why this population is not in linkage equilibrium. This is in linkage disequilibrium. So this concept of LD was described by Jennings in 1917. Now let's quickly see what is haplotype. Haplotype is a stretch of genome that tends to inherit together or ordinarily does not undergo recombination. A stretch of genome, a stretch of nucleotide within which there won't be any recombination occurring, that will be called as haplotype. And we'll be talking about a single individual, that is called haplotype. So as I said, the linkage disequilibrium can be between two locus. So if this LD block is larger, the haplotype will be larger because recombination is not occurring. That's why the entire stretch will be called as haplotype. So as the larger the LD block, haplotype will also be larger. And because it is larger, there will be fewer number of LD blocks per genome. It will require less number of markers to identify them, and the QTL resolution will be poor. Why QTL resolution is poor in case of large LD blocks? As the LD block is larger, we are not able to go closer to the QTL or go closer to the gene. That's why the QTL resolution is very poor. But the power of QTL detection is high. But when the LD blocks are smaller, the haplotypes will be smaller because there is recombination. So haplotype will be smaller. There will be large number of LD blocks per genome, and it will require more number of markers to detect those QTLs or LD blocks. And QTL resolution will be high because we are approaching closer to the gene. Because recombination is happening, we are going closer to the gene. That's why the QTL resolution will be high and the power of QTL detection will be low. So if we have two locus, how to estimate whether there is linkage disequilibrium between them or not, whether there is LD or not. So let's see an example. We have locus 1, locus 2. We have out of 8, we have 4 individuals carrying capital A, capital B. These are different alleles, capital A, small a, and capital B, small b. So 4 individuals are carrying capital B, capital A, capital B. 3 individuals carrying small a, small b, 1 individual carrying small a, capital B. So we need to calculate the genotypic frequencies as well as allelic frequencies. So genotypic frequency of capital A, capital B, so it will be 4 out of 8, so it will be 0.5. Small a, capital B, it is 1 out of 8, so it will be 0.125. Then small a, small b, it will be 3 out of 8, so it will be 0.375. And capital A, small b, it is not there, that's why it will be 0. Likewise, we need to calculate the allelic frequency. So frequency of capital B will be 5 out of 8, so it will be 0.625. Or we can simply sum up these two values and we can get the allele frequency of capital B, small b, and if we sum up these, we can get small allele frequency of small a and capital A. After calculating these, we need to go for LD estimation. So Levontin in 1964 has given one measure for LD estimation, that is D, which is equal to genotypic frequencies minus respective allele frequencies. So if we take capital A, capital B, the genotypic frequency is 0.5 minus the respective allele frequency. So capital A, this allele frequency is 0.5 into capital B, 0.625. So answer will be 0.87. If D is equal to 0, there won't be any LD between those two locus. If D is other than 0, then there will be an LD. But here we have taken capital A, capital B. If we take capital A, small b, the value will be same, but there will be negative sign before the value. So it will be minus 0.187. So there is nothing like negative or positive LD. It can be either present or absent. So to remove this confusion, another measure was reproposed, that was D prime, which is equal to D by D max. We know how to calculate D. What is D max? D max is if the D is equal to 0, as in this case, D max is minimum value of any of these two values. So allelic frequency of capital A and B will be less in this case. So this will be taken in the calculation, and when we calculate, it will be 1. So if it is 0, D prime is 0, then LD is not there. If it is other than 0, LD is there. Another measure is there given by Hill and Roberts in 1968, R square. R square is coefficient of correlation, which is squared, which is equal to D square divided by all the allelic frequencies. When we calculate this, we'll get the extent of LD. So as I said, LD can be estimated between two locus. By locus, I mean the markers that we are using. So suppose we are using 10 markers. So we can estimate LD between all the possible pairs of those 10 markers. For example, between 1 and 2, 1 and 3, 1 and 4, or 2 and 3, 2 and 4, likewise. All the possible pairs, we can estimate the LD. And when we plot this R square value against the genetic distance, R square value against the genetic distance, then we can get the LD decay plot. So why this LD decay plot is significant, where we can use it? It can roughly gives us the idea about size of the LD block. So if we draw a horizontal line from point 1, then and we can drop down from the intersection point, so it will be around 36 or 37 centimorgan. So in this particular case, the LD block, size of the LD block will be around 36 to 37 centimorgan. So using this, we can decide the number of markers that we are going to use. If the graph comes like this and the LD block size reduces, if it comes to 10 centimorgan, then we need to use large number of markers because LD block is getting reduced. So this is how this LD decay plot is important. This is kind of representation for LD. Another representation is a triangle plot between pairwise LD. So horizontal and vertical axis, there will be markers will be plotted, and upper triangle will represent the R square value that we have calculated using R square estimation, and it will be represented by different colors. Then the lower triangle will represent the respective p-value. P-value is significance. It will represent the significance of those R square values. So this is kind of visualization, how we can represent the LD. So till now we have seen association mapping, the basis of association mapping, that is LD, how to estimate LD and how it is represented. Now let's see how we can conduct an entire GWAS experiment. So first of all, what are the requirements? we need a germplasm panel. Then we need to target the uh, traits for phenotyping. We need to go for phenotyping of those traits. Then genotyping platform. By genotyping platform, I mean that we will be using the SNP markers in most preferably in GWAS, because we have to cover the entire genome and we need to get maximum number of markers which are polymorphic. That's why we'll be using SNPs. Then after that, we need to go for statistical tests to understand the structure of the population and identifying the marker trait associations. So first is germplasm panel. We can use natural populations, land traces, or germplasm collections, germplasm bank collection, core collections from gene banks, synthetic populations, or elite breeding material. We can collect entire elite breeding material from different countries and we can directly use it. We need to see that there is there is a large extent of phenotypic and genetic diversity. After that, we need to phenotype them. We need to identify the target trait for the phenotype. We need to phenotype them, and to avoid the minor gene effect, we need to go for multi-location and multi-year testing of those genotypes for that particular trait. But because we are using here high number of genetic accessions, normal complete block designs or RBD will be difficult or will not work in this case. So we'll be using incomplete block designs like augmented RBD or alpha lattice. Then we can also use the high throughput phenotyping technologies, which will be an added advantage for phenotyping. We can go for lab phenomics, field phenomics, or drones in this phenotyping. In case of genotyping, because the rate of sequencing per data point is reducing down, we can use advanced genotyping technologies like genotyping by sequencing, RAD sequencing, DART sequencing, or we have genotypic arrays like Affymetric chips and Illumina chips. We can also go for whole genome resequencing. What is this whole genome resequencing? This is actually a costly affair. So institutions from all over the world, they come together and they do whole genome resequencing of large number of accessions or genotypes, and they upload the data, genotyping data on the internet. So this is one project, the 3000 Rice Genome Project. They have resequenced whole genome of 3000 different accessions of rice, and this data is available freely for all the users, and we can simply download the data. We can indent the accessions, those we need from the IRRI, and we can go for phenotyping, and we can use this free genotypic data for GWAS analysis. That's how this WGRS will be helpful. Now, after having a very good phenotype and very good genotype data, we have issues of false discovery. Why this false discovery will occur? The associations that we are finding, those will be false. Why this happens? This happens because relative over representation or under representation of allelic frequency within the populations. So what is the reason behind it? The reasons are, first is hitchhiking effect. When there is any favorable mutation in case of a trait, the frequency of that favorable mutation will increase with time, and the linked markers, linked genes, frequency of those linked markers and linked genes will also increase with the favorable mutation. So that will cause over-representation of that allele, and that will lead in false discovery of association. Another case is familial related. If two individuals are related because of common ancestry, the alleles will be same, and that's why it will be overrepresented. The structure in the population. Structure means, suppose in case of rice, we have collection from indica varieties and japonica varieties. So certain alleles are having high frequency in indica, certain are having high frequency in japonica. Certain alleles will be similar in both the cases. So when we take all these accessions together as a germplasm panel, the there will be relative over representation or under representation of certain alleles, and those will cause false discovery. This geographic isolation is same as that of structure population. And epistatic selection, when there is association between a trait, a gene, and a marker, but this gene is another having again having epistatic effect on gene 2, then this SNP will automatically get associated with gene 2 as well. So that is not actual association, but because of epistatic effect, it is a false discovery. So how to negate this structure population or relatedness from the uh, analysis? We can go for analysis of structure of the population. To analyze the structure of the population, we use a software called structure, and it will divide the mapping panel into different subpopulations represented by different colors. Each line represents each individual or individual accession, and the output of this analysis will be a Q matrix, which will be generated by structure software, and thus this Q matrix will be used as an input in case of association analysis. We can study the structure of the population using principal component analysis as well. We can see here different colors are represented. So these different colors represent a subpopulation within the germplasm panel. Then next is familial relationship. So as I said, familial relationship, it is because the common ancestry, the allelic frequencies will be overrepresented. So to study this familial relationship, we have kinship matrix. We have software like GAPIT software, which will give a kinship matrix. As in population structure, it gives Q matrix. Here it will give K matrix, which will be used in association analysis. So we have phenotypic data, genotypic data, Q matrix as a population structure, and K matrix as familial relationship. So after having all this data, we need to select the method for GWAS analysis. So there are three methods: generalized linear model and mixed linear model, then multi-locus mixed linear model. So this GLM, generalized linear model, uses only genotyping phenotypic data. Then MLM uses genotype phenotype and Q and K as well. And the MMLM uses Q, K, and genotype phenotype as well as the background genotype. So in case of GWAS, we will go mostly for mixed linear models for association analysis. So this is the mixed linear model. Phenotype is equal to effect of background SNPs plus effect of target SNPs plus effect of structure of the population plus effect of kinship plus error. So when all the values for a particular trait and SNP fits into this model, then that particular SNP is said to be associated with that particular phenotype. These are the different programs we can use for association analysis. GWAS program is Tassel, which is used for LDDK plot, drawing LDDK plot, then analyzing the association. Then we can use structure for estimating the structure of the population. GAPIT and NAM can also be used in our platform for association analysis. So this is a snapshot of entire GWAS experiment. So we have phenotypic data, genotypic data. If we use only these two data in analysis, the false positives will be highest. If we include structure of the population and kinship data, we can see that false positives are reducing down. So after doing this association analysis, how these associations or marker trait associations are represented? Those are represented using QQ plot or Manhattan plot. So QQ plot means quantile plot, where expected minus log of expected p-values are plotted against minus log of observed p-values. So this red line indicates expected p-values, and these dots, these are individual SNPs, represent the observed p-values. So if there is deviation from these expected values, those SNP are said to be associated with that particular trait. In case of Manhattan plot, the markers are distributed on horizontal axis. We have large number of SNPs, so according to chromosome, we are plotting them on the horizontal axis against minus log of p-value. So in case of our study, we'll be using 0.01 as highest significance, p-value as highest significance. So minus log of 0.01 will be 2. So all this will be used as threshold. So all the SNPs above this line will show are supposed to be associated with a particular trait. So, but in this case, we can see there are large number of SNPs that are showing association. So these are actually false associations or spurious associations. Why this happens? Because we have large number of markers. So what is the significance? 0.01. So 1% chance of 1% error is there. So if we are using suppose 50,000 markers, so 1% of 50,000 will be 500. So there is chance that 500 SNPs will show wrong association. So for that correction, we need to go for Bonferroni corrections, where p-value will be divided by number of markers, and the value will be 6.7. So all the values, all the SNPs above this 6.7 will be actual associations. We have seen the entire procedure of how to conduct the GWAS, how to analyze them, and how it is represented. Now let's see the case study. GWAS, first case study is about GWAS to identify genetic loci for resistance to yellow rust in wheat in pre-breeding lines derived from diverse exotic crosses. So material used here is 419 pre-breeding lines, which are derived from crossing of 15 best elites from CIMIT. And the phenotypic evaluation was done for yellow rust resistance using alpha lattice design with 2 replications at 5 locations. And artificial production was done using suspension culture of Buccinus triformis, which was injected to the susceptible cultivar Morocco, which was planted as hill plots and border rows. And the data was taken for 5 different locations. Then the principal component analysis, kinship, and association analysis, Manhattan plot, all these were performed using the Passel software, population structure using fast structure software. Genotyping was done using 22,415 SNPs. And after the analysis, they have found that in all the 5 locations, the phenotype, the yellow rust resistance, has shown normal distribution, which denotes the polygenic response of the cultivars. When they combine the all the data of 5 locations, even then the normal distribution was found. The structure software divided the panel in 3 different subpopulations. Even the PCA study showed there are 3 different subpopulations. When they studied the LD-decay plot, they found that A genome, the LD decays at 2.5. For B genome, LD decays at 5 centimorgan. And for D genome, LD decays at 2.5 centimorgan. When they combined these 3 genomes, the LD decayed at 2.5 centimorgan. So this gave a rough idea that size of LD block is 2.5 centimorgan. In the Manhattan plot, after association analysis, they found that these 14 SNPs represented in top plot are common in all the 5 locations. So these were said to be significantly associated with the trait, with R-square value ranging from 6 to 14.2%. And these were the SNPs. Numbers represent the SNPs that were found. So they have classified these numbers according to the presence of these SNPs within the 2.5 centimorgan range. As I said, LT block is 2.5 centimorgan. So they have classified if the SNP is within that 2.5 region, then all the SNPs are supposed to be originated from single QTL. Accordingly, they have classified them, and they have found 7 different QTLs on 3 chromosomes for yellow rust resistance in wheat. Another case study, genome-wide association mapping to identify genetic loci for cold tolerance and cold recovery during germination in rice. Material used was 257 rice accessions, which was collected from different subspecies and collected from different parts of the world. The phenotyping was done in growth chamber in controlled dark condition. CRD was used in 3 replications, where 30 to 40 seeds were used per replication. These were different parameters used for the phenotyping: low temperature germinability, 13 degrees Celsius. For 28 days, germination was tested, and germination percentage was recorded. Germination index, the control here was 30 degrees Celsius for 7 days. So again, the values were normalized with the control, and the normalized values were used for germination index calculation. Then coleoptile length under cold stress was measured. Then after 4 days of recovery at 30 degrees Celsius, the plumule length at 4 days recovery was calculated. When they divided by 4, the plumule length recovery rate was obtained. Genotyping was done using 5,185 polymorphic SNPs. Population structure was found using structure program. GWAS analysis was done using GAPIT software and Tassel software, and Manhattan plot was drawn using R software. The structure software divided the mapping panel in 9 different subpopulations, and the diversity of for these particular traits, all the traits, is represented by the box plot. So we can see box first represents the very high diversity among the within the population as well as entire mapping panel. Then they found out.
5 more minutes left, Shivaji. You should wind up in 5 minutes.
Yes, ma'am. Yes, ma'am. Large number of SNPs they found for different traits they have under study, and these were the different QPS they found. So after seeing this case study, we can say that using GWAS we can pinpoint a particular SNP for a particular trait. Now, what after identification of that SNP? We need to go for identification of genes. We can annotate those SNPs and genes, and we need to validate them through expression profiles. We can validate them using biparental mapping population. We can design the markers using those SNPs, and we can use them directly in marker assisted selection, and further cloning and functional characterization has to be carried out. What are the limitations of GWAS? So most important is we cannot analyze the rare variants in case of GWAS. We require dense marker coverage, that's why it is a little expensive. We cannot study epistatic QTLs in case of GWAS, and power of QTL detection is very low. In future prospect, I will say if we include the genomics technology in phenotyping, it will give more accurate results. We need to go for more algorithms where we can analyze the rare alleles or rare variants. Bayesian recurrent method can be used to overcome the genome-wide error rates, and we can produce MAGIC and NAM population. Which can combine benefits of linkage mapping as well as association mapping together. And if we have whole genome resequencing in most of the major crops, it will be an added advantage. With this, I would like to thank you.